Hi, welcome you all to our science class. Today we are going to see about hormones. First definition. So hormones are chemical substance produced in the body that controls and regulates the activity of certain cells or organs. So hormones are the chemical substances which are produced in our body, and the role of the hormone is to control and regulate the activity of certain cells. So the activities like digestion, metabolism, growth, development and reproduction are controlled and regulated by these hormones. Hormones are produced in both plants and animals. The hormones which are produced in plants are called plant hormones or phytohormones. So plant hormones are also called as phytohormones. Here we are going to see about plant hormones. First we can see the types of plant hormones. There are 5 types of plant hormones. They are auxins, cytokinins, abscisic acid, gibberellins and ethylene. These are the 5 types of plant hormones. You can keep this in your mind easily by the word A cage. So A C A G E cage. A for auxin, C stands for cytokinins, again A stands for abscisic acid, G stands for gibberellins and E stands for ethylene. So these are the 5 types of plant hormones. First we can see about the plant hormone auxin. So this auxin was the first discovered plant hormone. In 1880, Charles Darwin and his son Francis Darwin, they observed that there was an influence which was transmitted from the tip of the coleoptile to the basal region. So these two persons, the father and the son, they observed that there was an influence. That influence is transmitting from the tip of the coleoptile to the basal region. And this influence was isolated and discovered by Wendt, and that influence was the chemical substance auxin. And this term auxin was introduced by Kogel and Hagen Smith in 1931. So Wendt discovered and isolated auxin. So how he discovered this auxin? So we can see about that here. So the Wendt, scientist Wendt, his full name is Fritz Warmolt Wendt. His period is 1903 to 1990. He did his experiment in the coleoptiles of Avena sativa. So he did his all experiment in the coleoptiles of this Avena sativa plant, which is commonly known as oats. So what is coleoptile? So coleoptile is a sheath which is protecting the young shoot tip in a grass or cereal. So the young shoot tip of the grass or cereals were protected by a sheath, and that sheath is known as the coleoptile. And in that coleoptile only this Wendt did his experiment. This is the Avena sativa plant, that is the oats plant. In this plant only Wendt did all his experiments. So now we can see the different experiments of Wendt for discovering this auxin. So in his first experiment, he removed the tip of the Avena grass and he noticed that the plant did not grow, which indicates that the tip producing something which is essential for growth. And in his second experiment, he removed the tip of the grass and he placed the agar block on the decapitated end of the grass. This also did not show any response. So in his first experiment, just he removed the tip of the grass and he noticed that the plant did not grow. And in the second experiment, he removed the tip of the grass and he placed the agar block on the decapitated end. Here also the plant did not show any response. And finally, in his third experiment, what he did is, he removed the tip of the Avena grass. After removing that tip, he placed the tip on the agar block and he allowed the tip to stand on the agar block for 1 hour. After 1 hour, he discarded the tips and then he placed the agar blocks on the decapitated coleoptile. And then he observed that plant starts to grow up straightly towards the sunlight. We can see the diagrammatic representation of Wenn's experiment. So in his first experiment, he took a coleoptile, then he removed the tip and which does not show any growth. In his second experiment, he took a coleoptile, he removed the tip and then he placed the agar block on the decapitated tip. So the agar block contains nutrients, so he kept that agar block on the decapitated tip. It also did not show any growth. In the third experiment, he took the coleoptile, he removed the tip and then he placed that removed tip on the agar block, and after 1 hour he discarded that tip and placed that agar block on the decapitated tip. Then he observed that the plant started to grow towards the sunlight. All these experiments indicates that some chemical had diffused from the cut coleoptile tips into the agar block. And then Wentz concluded that a chemical has diffusing from the tip of coleoptiles was responsible for growth. That is, he is saying that a chemical is getting diffused into the agar block from the coleoptile tips, and that is responsible for, and that chemical is responsible for growth, and he named that chemical as auxin. It means to grow. So the meaning of auxin is to grow. Here we can see the types of auxin. There are two types of auxins. They are natural auxin and synthetic auxin. Natural auxins means auxins which are produced naturally by plants. And synthetic auxin means artificially synthesized auxin. But they have the properties of auxins. Synthetic auxins means these auxins are artificially synthesized, but they have the properties of auxins. Examples of natural auxins are indole 3 acetic acid, phenyl acetic acid, IAA and PAA. Then the examples of synthetic auxins are 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid, alpha-naphthalene acetic acid, indole 3 butyric acid, indole 3 propionic acid. These are all about the types of auxins and its examples. Here now we can see about the physiological properties of auxins. Auxins promote cell division. So, due to cell division only, plants or animals, anything can grow. So, in plants, auxins promote cell division and helps the coleoptiles to grow. It induces root formation at lower concentration and it will inhibit it at higher concentration. And the very important function of auxin is apical dormancy. It means auxins are produced by apical buds. But this auxin will prevent or inhibits the development of lateral buds. The auxins are produced by apical buds, but it will prevent or inhibits the development of lateral bud, that is apical dormancy. Then these auxins helps in the fertilization of parthenocarpic plants. Parthenocarpic plants means the seedless plants. Example, tomato, pineapple, watermelon, grapes, etc. These are the examples of parthenocarpic plants. So in parthenocarpic plants, fertilization was induced by auxins. Then these auxins will prevent abscission in premature leaf and fruit, and it promote abscission in old leaves and fruits for falling. So because of this auxin only, these premature leaves and fruits are not falling down, and this auxin only helps the old leaves and fruits for falling down. So these are all about the auxins.